on CA simulation of RNA self-replicators. I have a very hard time saying the word cellular automata. Whenever you're ready.
Thanks. Just to check, you're getting my screen now, right? Yeah. Okay. So yeah, thanks for the introduction. So I'm Ian Holmes. I'm going to talk about RNA biophysical models. From my personal perspective, Alife is kind of at the intersection of my background as an 8-bit game developer and my career in bioinformatics. So pronunciation issues aside, I know I'm probably not unique at this conference in my love of cellular automata. I really love them. And so these game prototypes started from a collaboration with Maxis on Spore 2, the sequel to Spore, which was sadly cancelled. I think it's long enough after it that I can now waive the NDA and talk about it. But basically, I made several prototypes trying to explore various game mechanics around CAs. I tried sort of entropy balancing. I tried various kinds of God games. I even prototyped a little cellular automata game that's sort of a snake game called Nematode, and you can play a solo version at nematode.de, this URL here. If you want to zone out while I'm talking and play that, I'd be perfectly happy for you to do that. I'm actually not going to talk about games today. If you're interested, I have a talk from the Roguelike Convention in 2017 that is on YouTube. The connection is that in a few of these games, I built a functioning model of RNA folding kinetics, which is what I want to talk about today. And so this is motivated by my academic interest in molecular evolution and developing bioinformatics algorithms for RNA folding. So the RNA world, of course, is this idea that self-replicating RNA molecules are the ancestors of life. And this idea sort of came up in the 60s. Carl Loza and various others, motivated by observations of Crick and so on. So over time, circumstantial evidence has built up to support this idea. So for example, the known biochemical and functional repertoire of RNA has expanded with various discoveries. For example, these small molecule sensors, riboswitches, discovered by Ron Breaker to occur naturally after he had created some synthetically by in vitro evolution. Crystallographic data has confirmed what many biologists had intuited, molecular biologists, that the functional core of the ribosome is pure RNA. The machine is an RNA machine with just protein adjuncts on it. And of course, there are theories, various theories of how self-replicating RNA arose. For self-replicators to thrive and to multiply, it seems they need to be locally concentrated, for example, inside micelles, which of course are essentially soap bubbles that form spontaneously from lipid bilayers. So this is a movie from Janet Iwasa, the visualization pioneer, who was working at the time with RNA World legend Jack Szostak. And within these bubbles, the self-complementary nature of nucleic acids means that an RNA molecule can be a scaffold and a catalyst effectively. It enables the formation of a complementary sequence. So if you have a sort of fuel substrate, if you have single nucleotide monomers in the mix, then it can essentially copy itself spontaneously. So in order to sustain life, RNA probably has to catalyze a few other reactions too. And this is where the accumulated discoveries of the past few decades are very important in lending credibility to this thesis. So I did mention, for example, the small molecule sensing riboswitches. There is peptide formation at the ribosome. More recently, biologists have discovered RNA ribozymes, RNA enzymes that perform redox reactions. This was kind of one of the last nuts to be cracked. And then we also know that RNA can, in fact, cleave and form the phosphodiester bonds of its own backbone, as in this hammerhead ribozyme, for example, which is found in plant viroids, and various other kind of self-cleaving RNAs like self-splicing introns, which arose. There's sort of a whole evolutionary debate about at what point they arose in evolution. But they're essentially introns that sort of splice themselves out of genes. And so this hammerhead ribosome actually was used in an early example of an RNA logic gate. And I did want to just mention this briefly due to the Alife and AI connection of this meeting. So nucleic acids have been used in nanotechnology and molecular computing in various ways. So if you make various carefully chosen synthetic assemblies of DNA and RNA sequences, you can build logic gates that operate basically by reversible reactions involving strand displacement. So no covalent bonds are being broken or formed, only hydrogen bonds. So this effectively is all a sort of reversible reaction. It's quite efficient. And this is an example of an AND gate. This is Eric Winfrey and others who have developed these. And essentially by introducing the right RNA, the right DNA strand, you kind of cause an allosteric disruption of this existing complex, and it forms a different complex, and in this case releases a fluorophore that is sort of the output of this gate. And if you string enough of these together, you can build quite complex liquid circuits like this perceptron from 2011. So Microsoft Research has really capitalized on all this, has made a compiler that will convert circuit diagrams or conceptual representations of Boolean circuits to DNA sequences that will implement these circuits as gates. They have a team working on DNA computation. So I just mentioned this because of the Alife and AI theme, but it also is part of why I'm interested in working on these sorts of nucleic acid models in particular, because there are cool things that you can build with them materially. So, you know, tests of Alife can be computational simulation. So as with Tim Hutton's work on artificial cells, which recapitulate many of the sort of, in somewhat abstracted form, many of the things that I'm aiming to model, and we'll talk about here. Or they can be experimental systems like Jared Joyce's self-replicating RNA complexes, which as fuel basically have sort of smaller polymers that are subunits of the main complex, but they can then copy those. Or they can reassemble themselves using those things. So if we think about sort of simulations and computational Alife, we can imagine a spectrum from abstract to biological, I guess, with Automata at one end, like von Neumann's universal constructor, or these various machines of Stevens, which can be found on YouTube. Some very impressive self-assembling machines. And then you get these increasingly sort of plausible, as you move across the spectrum, these sort of artificial chemistries spanning various kinds of protocell and ribocell. And so these RNA molecules or RNA models that are actually simulating RNA, I would say are up at sort of the biological realistic end of the spectrum. It's not a value judgment, they're just kind of more based on actual material things rather than sort of pure virtual things. I'm drawn to that end by the possibility of computational discoveries that might actually be synthesizable, or, you know, even as in the case of Ron Breaker's riboswitches, discoverable in nature. So if you can come up with an idea, that maybe you can find it in the many genome sequences that are out there. So in bioinformatics, when we think about RNA structure, we often think about it quite abstractly as a sort of a graph of base pairs. So in this kind of view, the graph view, the spatial coordinates are effectively eliminated completely. It's just about which bases are in contact with which other bases. And so this is an efficient sort of abstraction, and it means that dynamic programming algorithms can be used to predict the structure, but it does miss some nuance. So more recently, these more coarse-grained models have been developed, and several of these use a sort of what's called a two-tolerant lattice. So basically the RNA polymer in these molecules is a random walk with one nucleotide per step. It's a self-avoiding walk except where two bases are paired, where they can then occupy the same cell. And so my Carnival program that's described in the proceedings and is available as GitHub URL uses a two-tolerant lattice on a cubic grid with a simple energy model that basically rewards hydrogen bonds, rewards bases with more hydrogen bonds, and it rewards anti-parallel stacking of bases. But it doesn't include bond torsion angles, it doesn't include more detailed energetics of base pair interactions. So it's pretty simple. The monomeric units can diffuse by making steps to adjacent cells on this lattice, and it's modulated by a sort of Metropolis Hastings accept reject possibility, a probability based on the energetics of the base pair model. Compared to previous RNA models, this is actually very simple compared to the model of Justin Everez, which I introduced the two-tolerant model with, for example. This is very simple. And yet it does actually recapitulate aspects of RNA structure. So in this example of a yeast transfer RNA for phenylalanine, The experimentally determined structure has a pseudoknot, which is a base pairing interaction that can't be predicted by the very simplest of the RNA folding algorithms. It's a little tricky to predict. The lattice folding model gets at least part of it right. It finds some of the approximate location of the pseudoknot, if not every detail. So it's a plausible model, not totally strict. And then to this model, we add very simple template-driven polymerization kinetics. So basically, we allow two adjacent bases to form a covalent bond if they're held in place by a template strand. So if two monomers diffuse to adjacent places and they're held by a complementary strand, they will then form a bond. And this is enough to generate self-replication behavior. So the fidelity of this replication depends a lot on the probability of mismatches, which is part of the energy model, as you might expect. We also observe naturally occurring circularized molecules, which we didn't anticipate in the design. They weren't planned for. So you naturally get things that look a bit like plasmids, circular nucleic acid sequences. And in fact, the lack of torsion energies in the bond, in the energy model, so the lack of sort of bond angle models, that means that some of these circles have an unrealistically tight turning circle, and you get plasmids of just sort of 3, 4, and 5 nucleotides, which wouldn't occur in nature. In nature, there's sort of a high entropic and energetic cost to making a circle that tight. So that's room for refinement in the model there, but you could still kind of maintain a cellular automata model, and that was quite important to me in trying to design something that would sort of fit with some of the other dynamics that we want to put into this. Particularly, these early results basically don't have any model of micelles or soap bubbles, but I'm particularly interested in seeing if we can kind of combine this quasi-realistic model for RNA folding and replication with similar sort of coarse-grained models for the amphiphilic lipid bilayers and micelles, which represent, for example, in this paper by Bogosian et al., represent the lipids as single particles on the cellular automata, but with a dipole moment that allows them to kind of stack up in these lipid bilayers. So with that, I'm going to thank you for your attention, and also thank some sources of funding and inspiration, the NIH and UC Berkeley, and Rudy Rucker in particular, the author and cellular automata wizard, who's a friend and inspiration. Richard Evans of DeepMind, who was my Spore collaborator and is constantly an inspiration in terms of artificial life, and also Chris Mungle at Lawrence Berkeley Lab. So I want to thank you and thank the organizers very much.
Thank you, Ian. That was great. Awesome talk. I'm curious to see what questions the audience has for Ian. You can use the chat or raise your hand. I think I'll get the conversation going. I feel like I either missed it or I had a preconceived notion of what I expected the talk to be. So you call this a cellular automata or a CA, and does that mean that you sort of initialize the system with the right base pairs for like an RNA structure that you want to try and replicate, and then you have local rules, like deterministic rules that tell you how it can organize?
Yeah, well, it's a cellular automata in that it is all built up from local rules. The cellular automata states are essentially individual RNA nucleotides or nucleotide pairs on this two-tolerant lattice, and they. But because they're allowed to have bonds to adjacent pairs, then you effectively are allowed to have polymers within this CA. And so a lot of the things that I've been, working with, the various kind of toys that I've played with, and I'm just going to share the screen again very quickly to show you this. These sort of games and toys in particular, you can see these ones in the top bottom left and the bottom right are actually using these sorts of polymer models, and they are technically cellular automata. They all just involve the Moore neighborhood essentially. So it's just the 8 cells around any given cell on the square lattice. But by kind of poking registers into those cells, they can maintain bond integrity, and so you can get these kind of polymeric structures which are sufficient to then construct these sort of micelles and so on. So, so this, I mean, I didn't go into it too much, but you get quite a lot of fun game mechanics out of this, where you can kind of build these kind of flexible ropes and tethers and things like that, and bubbles and so forth. But you can also then sort of simulate all these sort of these biogenesis kinetics in these sorts of systems.
Very cool. I see, Hiroki, you
yeah, actually, I saw that Christopher asked a question in the chat, so I give the the time for Christopher.
So Christopher is asking if you could please say again what two tolerant means and clarify whether the CAs are deterministic.
The CAs are not deterministic. These are stochastic cellular automata. Of course, they're stochastic CAs running off a pseudo-random number generator, so you could also think of them as being deterministic, just with some kind of pseudo-randomness built in. The idea of a two-tolerant random walk is a random walk which is so you have a random walk that's sort of self-avoiding is one that's not allowed to double back on itself. A two-tolerant random walk is one that's allowed to double back on itself only once. So you can have not just one step in every cell, but you can have two steps in every cell. And with the RNA ones, you effectively then further restrict it so that those are only energetically favored if those two steps sharing a cell are complementary bases. So this means that you can kind of coarse grain an RNA molecule and get quite efficient folding dynamics, and actually explore RNA structure space in a reasonable amount of time, which is necessary to get these sorts of replication kinetics and also just cool visual dynamics and so on.
Very cool. I'll second Christopher that it is